June starts the period of having a hard time of getting a crowd, so I appreciate everyone who came. If you haven't registered or signed in up there, you can scan this QR code, sign in online. Really helps us when we do our reports to be able to list how many members we've had at the meeting versus non-members. So if you haven't done that, please do that. A couple of topics I wanted to cover. This will be our last meeting for the summer, and we'll start again. When is your August?
Next year talk should be in September.
September? Okay, so yeah, our next full next presentation will be in September. So be watching this summer for that announcement to come out. A couple other topics. If you're interested in volunteering, you can always reach out to me or to Paul or Lena, or you can go to our website and access contact info through there. So please check that out. Elections are coming up this year. So we're hoping to kick off the elections pretty soon, which means that we need anyone who's interested in being on the board to announce that they're interested in being a candidate. So send us an email if you have any interest or know someone who is interested in being on our board, so we can get them involved in the election cycle. That is about all I have for tonight. I hope everyone has a good summer, but for tonight, we turn it over to Richard, who's going to introduce our speaker.
Thanks, Chris. And again, thanks everyone coming in person and everyone is on their Zoom. So today we're very glad to have Professor Scott Acton from University of Virginia. We'll continue our distinguished lecture series, and this time we co-hosted with IEEE Minnesota section. So let me introduce our distinguished lecturer, Dr. Scott Acton. Dr. Scott Acton is Lawrence R. Collier Professor and a Chair of Electrical Computer Engineering at the University of Virginia, as well as AI advisor to the provost. He is also appointed in biomedical engineering. He was a program director and acting deputy division director in the computer and information science and engineering directorate of the National Science Foundation, where he was given the Director's Award for Superior Accomplishment. He received the MS and PhD degree at the University of Texas at Austin and a BS degree from the Virginia Tech. Professor Acton is a fellow of the IEEE for contributions to biomedical image analysis. He is also fellow of American Institute for Medical and Biomedical Engineering, fellow of the Asia Pacific Artificial Intelligence Association, and a fellow of Artificial Intelligence Industrial Alliance. Professor Acton's laboratory at UVA is called VIVA, Virginia Image and Video Analysis. They specialize in biomedical image analysis problem. Professor Acton has over 400 publications in the image analysis area, including the books Biomedical Image Analysis, Tracking, and Biomedical Image Analysis Segmentation. He was 2018 co-chair of the IEEE International Symposium on Biomedical Imaging. Professor Acton was editor-in-chief of the IEEE Transaction on Imaging Processing 2014 to 2018. So let's welcome Professor Acton.
Thank you, Richard. Thanks, Joel. How do we
Switch the slides over.
Oh,
It's my first time in Rochester. I'm assuming that the weather is always like this.
Sure.
I took a 21-mile bike ride around the town this morning, and very bike-friendly city. A lot of beautiful pieces of nature, shall I say, the Cascade Creek and the lake, and going around the river. And there's construction everywhere, so that must mean that this is a city that's growing and successful. So the talk title is probably a little overhyped, even though the AI revolution is underhyped, according to Eric Schmidt, one of the I guess the past CEO of Google.
Oh, I think it's just
Got it now?
down instead of.
Oh, cool. Thank you. Oh, did I hit the wrong one?
The down arrow?
Oh, down arrow. So I want to begin by telling you my life story. I was born in LA, and I can watch the back. It's cool. And basically, my family moved to New York, but we didn't like it. So we basically resettled in Northern Virginia, where I went to Oakton High School. I got my bachelor's degree at Virginia Tech down in Southwest Virginia, and got into my cheesy red Camaro, and there it is, and drove to Texas, where I got my PhD under Al Bovik, the Emmy award-winning Al Bovik at the University of Texas. Appreciate Motorola's support for my PhD. I never thought it would be cool to work in a fab until the last couple of years when we talked a lot about chips. My first academic job was at Oklahoma State. After a few years, I got back into a candy apple Ford pickup and drove back to my home state of Virginia, where I've been at the University of Virginia since 2000. That's my family. Colby on the left, just graduated from Purdue, and of course, electrical and computer engineering. My wife, Sabrina, my son, Henry, who's pre-med at the University of Virginia. I also spent 3 years, as Richard mentioned, at the National Science Foundation, and we can talk later over coffee or beer if you want to discuss what's going on there. I want to thank my lab and, of course, my support. We have support from the NIH, the NSF, a number of foundations, including Gates Foundation, for our education work, which we're not going to talk about tonight. That's the most recent picture of my lab over here. It's a little dark, but I'm going to acknowledge my collaborators through the talk, and I am the department chair, so I have to spend 1 minute bragging on my department. In 1 year, 5 of our assistant professors got the National Science Foundation Career Award, and we started calling them the Fab Five. Until later that year, 2 more got it, and so I guess they're the Fab Seven. But we have a lot of young, bright faculty that I'm proud of, and some established senior faculty as well. Today, what I'm going to do this evening talk is I'm going to ask 4 questions and explain 4 projects, 4 answers, based on questions that my collaborators, and in the last case, my student, Show Me, have asked. And they're all basically related to medical imaging. I realize that, it's dangerous to talk about medicine at the world's greatest hospital system, but I'm going to do it, even though I'm just an engineer and I know nothing about medicine. But I've been fortunate enough to collaborate with people like Yoni Kipnis, who knows a ton about medicine. And the question he asks, does the meningeal lymphatic system, which I will explain briefly, does it have anything to do with Alzheimer's disease? And how did image analysis play in that story? And then I'm going to talk about 3 more. So I'm going to talk about 4 projects. The risk is I don't go too deep into any one project, and engineers like deep. The benefit is if you get bored with one of them, you're probably only a few minutes off from the next one. So let's see. I'm going to ask 4 questions, explain 4 projects, and then I grew up in the evangelical church, so I'm going to end with a sermon on AI, on artificial intelligence. So you have that to look forward to. Some thoughts on AI at the end is all I'm saying. Okay, so first, my work with the Kipnis lab. Think about this. A decade ago, it was taught in medical schools, including this medical school, that there was no lymph in the brain. We all know what lymph is. These are lymph glands here. You know, we have lymph in our underarm. It's a waste disposal. It's a tissue that disposes of waste, the lymphatic tissue. Folks 10 years ago thought that all waste was taken care of by the spinal cord and by the vascular system. There was believed to be no lymph in the brain. Now, this group, Yoni Kipnis, who's now at WashU St. Louis, was at the University of Virginia at the time, discovered this lymphatic structure. It's actually in the meninges. It's in that 3-layer protection, spongy protection layer around the brain. And the question they asked was, could this play a role in degeneration? And of course, you all know more about medicine than I do. If it plays a role, then maybe there's a treatment, maybe there's a drug, maybe there's a process to address such degeneration. And this is something that we all have a relative that's dealing with. 1 in 3 seniors is dealing with degeneration in some form right now. So, I want to go back a second and explain a technique called level sets. Now, this isn't AI, so don't throw any tomatoes. I know that, you know, in 2025, you've got to talk about AI, and I'll get to that at the end in the last 2 projects. A level set method is, here's the idea. There's a function phi. Phi is like the height of your mountain. You're going to build this mountain that you're seeing on the left side, and you're going to grow with the differential equation, grow this mountain phi. We call it the level set function. Then we're going to cut it at a certain level, level 0. That will make a 2-dimensional contour, and that will be our segmentation, separating foreground from background. So the cool thing about level set methods is you don't need to know how many structures there are in the beginning. If you were segmenting coins, you wouldn't have to know how many coins there were. You wouldn't have to know the shape. But you basically control this function phi, and you build an energy function, or functional, that best preserves the topology, has certain properties, maybe smoothness. And this is what we use to segment the lymphatic system from confocal. And as I mentioned, this energy functional E basically is something we're going to minimize to come up with the best phi, this mountain, that fits this image. Let's call it f of x. All right. The only twist we did for the lymphatic system is we said, okay, there's a number of thin structures. They don't have much contrast with the background. We're really good at determining the background, so we're going to use a method called matting, which is from film. A method called matting to say, hey, this is definitely background. We're going to discourage segments from growing in the background. And basically, we came up with a way of finding these structures, which are sometimes very delicate and very small, and you know, on the order of on nanometer scale, submicron scale. And so this method you can find in the Journal of Physics, written mostly by my PhD student, Nazia. And that was the first step. The second step was, once we figured out the structure, was to figure out how waste was moving around. Now, it was a complicated problem. And basically, think of this variable, this phi, I'm using phi twice. This phi is the density of waste within a certain volume. If I throw flour, follow me here. If I make bread and I throw flour onto a pond, it will diffuse. And that's taken care of by this first term, a diffusion term. if I throw flour into the Cascade Creek, it's also going to move down the creek with a flow. That's advection, and that's this term. And then, of course, there might be sources or sinks, things where it disappears or we get new waste. What we were able to see is, okay, there's a chemical called Visidyne. It's used to ablate this lymphatic structure. You put it in the mouse brain, and the lymphatic structure melts, essentially. So it's no longer functional. With the Visidyne, this is the waste removal, according to our model, with the lymphatic structure broken. And this is waste removal with the lymphatic structure functioning. And so what they were able to do in the mouse model is basically come up with a model for how waste is removed in the brain, and then how that relates to AD, how it relates to Alzheimer's disease mice that you can, that can progress within months towards degeneration. And so this is sort of an anecdote, but it's an anecdote where image analysis, where electrical engineering really affected science and medicine. And it made the cover of Nature, this project, and we were very fortunate to be on it. The credit really goes to the medical doctors and the biologists for their wonderful science, but we were able to be a part of it by doing what I showed you. It was acknowledged by Nature Medicine and by Forbes, which was really interesting for us to be in the limelight. And basically, it's now believed that this lymphatic structure plays a role in AD, in Alzheimer's disease. How does it play a role? Well, I'm not the person to ask, but you can think about if waste is not removed, plaques can form, and you get the picture. So that was an exciting piece of research. I'm going to move on to another one that I worked on with my friend Barry Condren, and it's related. Think about at a microscopic layer, level. If you had pictures of two neurons, how would you compare them morphologically? How would you compare their shape? This might have also a role in degeneration. How do you quantify the amount of degeneration in a given neuron? And so we worked on this project over a number of years with Barry Condren. The context was the neuron. Maybe you remember that at first scientists basically described every one of 300 and, I can't remember if it's 302 or 304 neurons in C. elegans, the worm, maybe 302. And then next came the fly, the Drosophila melanogaster, the fruit fly. We get a lot of those in Virginia because we're in wine country for Virginia. And that's the model we were working on. And then, of course, the mouse is really the next goal, which people like Genelio Research Campus are working on in Northern Virginia. There's probably people here at Mayo as well. So here's our approach. It's graph theoretic. And it's different than, I think, than most previous graph theoretic approaches to describing neurons. We view a neuron, and this is like, think about this as our neuron. We view a neuron as a collection of paths from soma, cell center, to dendritic end. Forget about the axon for right now, if you know too much. So this neuron, we're going to say, has three paths. Root or soma to the end of dendrites. So it's a union of these three paths. And these three paths can all be parameterized from 0 to 1, start to end. And they can all be essentially, we can have their three-space position along that path. So we have that parameter. We also have two other parameters that I'm going to invent, so to speak. Those other two parameters are biologically significant, according to my collaborator, and they are, if we go down from root to dendrite, to the dendritic end, let's say, if we're right here, there are two paths that share this point. We call that concurrence, and that's my C term. We also have the notion of hierarchy. When there's concurrence, that's important. When there's larger hierarchy, that becomes less important. The morphology becomes less important. So hierarchy, what you could do is at this point, you could count the number of bifurcations above that point, and that would tell you 2, and we actually say number of bifurcations plus 1. So we have every point, we have 2 other terms, concurrence and hierarchy. And what we do is we make a little functional. We penalize you if you're different in concurrence. We penalize you if you're different than in location. More on that in a second. And we're okay if you're high in the hierarchy, the error becomes less prominent. And then what we could do is we can take 2 neurons, and we can auction the different paths as matches to one another using something like Hungarian algorithm. It's sort of a game theoretic approach. And then we can come up with an overall score for how well 2 neurons match. And maybe we could classify the neurons and say, this is not a motor neuron, this is a Purkinje, that sort of thing. But we can also do things like take mean between neurons, which is really fun. Here's a twist. And hopefully you can follow the idea. If we penalize the distances and position between 2 neuron paths, we could use Euclidean distance. We could use normal 3-space distance measures. However, that would be like a Procrustes analysis. Procrustes was this mean Greek that always made the bed a little smaller than the dimensions of his guest. But that would be like if we use this 3-space Euclidean distance going between the 1-legged man and the 2-legged man, this would be the progression in shape, which doesn't look very logical. What we decided is along with our collaborator, Anuj Srivastava, we decided we wanted to use an elastic distance. So we take all these points and we project them into a different space called square root velocity function. It's a real simple function. You can see it just uses the tangent. It's a function of the tangents at the various points along the curve. And basically, what this allows us to do is to do elastic distances. Basically, how much stretching and bending does it take to go from one contour to the other? And now you can see with the transition between the 1-legged man and the 2-legged man, and that looks pretty logical. And the same is true with midpoints between neurons. The elastic deformation proved to be really useful. Let me show you an example. This is a Purkinje neuron. It has something like 400 and something paths or dendritic endings. And this is, I'm gonna do generation of a neuron. So this was the work of Tamal Badable, who's now at MIT. Okay, so that's 2. I'm gonna go a little deeper into this one, but let's hit one more before that. And my colleague Natasha, who has been very successful as a young investigator, asked, can image analysis help in the assessment and planning of focused ultrasound treatment of essential tremor? This treatment, to me, is both fascinating and mysterious, and let me tell you a little bit about it. And maybe because here in Mayo, this is where all the cutting-edge treatments are being developed. Essential tremor goes like this. Essentially, 7 million Americans, that's a lot of Americans, maybe 1 in 40 or 50, are affected by this. You usually get it on one side. It will be difficult to brush your teeth. It will be difficult to eat. It will be difficult to sign a check. And so it really affects your quality of life. There's really, in just essential tremor, it's not like it's connected to other disease mechanisms. What Jeff Elias and his team at the University of Virginia have figured out is a treatment. It's really a surgery. I'm going to call it, we call it MRG FUS, magnetic resonance. So we use MRI to guide, and we're basically delivering energy by focused ultrasound, by focusing an ultrasound beam on a spot. It's a specific spot in the thalamus. It's what we call the VIM nucleus in the thalamus. And basically, you have two VIM nuclei. And a nucleus in the brain, as you probably know, is just a collection of neurons that that are dense and have a specific function. So basically, if you're having right-sided central tremor, they will ablate the nucleus on the left side, and vice versa. And this is all ultrasound guided. It's all MR guided, and we use CAT. We use the CAT scan to figure out the geometry of the brain before the surgery. And really what happens, it's amazing. I've seen this, you can see it on YouTube as well. 20 minutes later, people are writing so here's an example right here. The patient cannot keep it with him in the helix here, but right afterwards could perfectly do, better than I could, a trace to this helix, and that's a test they use. It's fascinating. It's a life-changing thing that occurs like in 20 minutes.
Quick
Yeah.
question.
Yeah.
The treatment, is that can be reversible? I mean, after treatment, the patient looks normal, but these symptoms could be reversible.
So the question from Richard is, could it be reversible? I wouldn't say reversible, but I will say that some patients need to be retreated after 3-5 years, and they're keeping data on that, and because the treatment is really, not as bad as some things could be. The patients are more than willing to go through it. There's two related research questions that we're working on. This is work in progress, so maybe you all can help me on this. The first is, okay, so we make these ablations, and you're seeing one right here. And basically what you're seeing is this area right here, the center area, is necrotic tissue, dead tissue. This area is area that's basically taking on water, edema. And so what we want to do is one day after this surgery, we want to use MR to find the size of the lesion and the position. And then what my student Samarth is doing is he's trying to figure out, does the size and position of the lesion relative to other geometric landmarks in the brain, does it have any correlation with side effects? And some of the side effects are verbal side effects, balance, coordination loss, numbness. And so we're investigating this. And we've at least got to the point where we think we do pretty well at segmenting these lesions. The second question is, and I find this sort of interesting. We all know that everyone's head is different. Everyone's skull is different. I have a different curvature, a different thickness than you have. And we know that affects the transmission of energy, the ultrasound. Can we basically use the pre-treatment CAT scan, CT scan, to basically plan the treatment better, knowing the thickness at various points and the curvature at various points of the skull? Because in ultrasound, what we can do is we can delay the different elements. We can add a phase correction and better deliver energy if we know what we call at various points the skull density ratio. Now, what you're seeing here is a high skull density ratio, and you're getting a great transmission of energy. Low skull density ratio, you're not. And this has been a global assessment. We want to look at perhaps doing a local assessment and a local variation for the treatment. And that's also work that Samarth, my student, is doing with Dr. Cheyenne Musa and Natasha Shabani, who now is his wife. All right, so let me show you some results from the first question. This is a segmentation of the lesion for this patient's brain. And essentially, what we're doing is we're using AI here, and we're using something called no new net You've probably heard of UNet because that's what everyone in medicine uses for segmentation. The authors of No New Net said, hey, we're going to come up for medicine, we're going to come up with a general segmentation tool and use transfer learning, which means use what a large body of imaging evidence of trained images to basically form as the basis for new segmentation problems, which we've learned, which we've adapted to in this application. We also actually segment the sinus, which is for to sort of get the position in the brain. And you can see this in this paper from a couple of years ago now, a year and a half ago, the method we used here. On the second question, this is evolving research, and that's why I say work in progress. These are two skulls from CT, and essentially what you're seeing here is the distribution and curvature values, which isn't dramatically different, but it's different. And my student points out in this patient, there's a very large dip near the temple that could affect transmission. And so what we're trying to do now is come up with a tool that does this computation automatically, and then come up with a method where we're going to modify the ultrasound based on curvature and thickness. We have various different thicknesses in the skull, and that affects this ablation process that we want to optimize. So that's work in progress, and hopefully you'll see more out of that, and hopefully that student will graduate. The last one I wanted to show you before I start into my sermon is from my student Show Me. It's also related to ultrasound, not necessarily restricted to the brain, which is the subject tonight, but this is definitely AI, and maybe you've heard of the diffusion network. I'm going to explain my version of the diffusion network as if you were in my class in 1 second. But the question she's asking is, okay, these diffusion networks are being used to enhance images. Maybe you've seen it. Can they enhance ultrasound imaging? And why would ultrasound be interesting? Well, it has signal-dependent noise. It has speckle, which are basically multiplicative in nature instead of additive. Can we use this type of network, which I'm going to explain, to enhance ultrasound, which is notoriously, noisy at least in the eyes of the patient? And I wanted to say before I started into this part that I'm not hopping on the bandwagon of AI. I've been on it for a while. So my dissertation was published in Transactions on Neural Networks. And what I remember from my advisor is he brought me in the room and said, Scott, it's okay that you did neural networks for a PhD, but never admit it. You'll never get a job. And so I just needed to wait 30 years to get a job. One of the themes in my sermon, which I'm going to hit here in a second, is AI is not a black box. AI is not a black box. I heard Sundar Pichai say on 60 Minutes that we think of AI as a black box, and I totally disagree with that. The Ford Mustang is a complicated piece of engineering. I don't know if anyone in here can explain every system of the Ford Mustang, just as you might not be able to explain every portion of ChatGPT, but we don't say the Ford Mustang is a black box. And we shouldn't, as engineers, say that technology that we're using is a black box. So let me give you my 2-bit description of a diffusion network. Okay, a diffusion network, first of all, is a cascade of autoencoders. What's an autoencoder? It's pretty simple. You give me a 3, and I'm going to encode that 3. The 3 has 700 and something pixels. That's 700 and something numbers. I'm going to encode it as 20 numbers. How would I do that? 20 neurons. And maybe those 20 neurons have 700 inputs. We could make it in MATLAB in 2 minutes. And so the encoder takes these 700 pixels and changes it into 20 variables that I'm going to call the latent space because it's hidden, because it makes no sense to us as humans. And then, because I have nothing better to do, I'm going to take those 20 variables and change them back into 700 variables and give you a 3 back. And the beautiful thing of this autoencoder is it needs no labeling. It doesn't need people in some country sitting in an 18-wheeler labeling images. Because all you're doing is you give me a 3, I try to recreate the 3. You give me a 4, I try to recreate a 4. It's an autoencoder. One twist on the autoencoder is the variational autoencoder. The only twist here is I'm going to say, okay, in this latent space, I'm going to try to enforce a distribution. Maybe it's a Gaussian distribution. Maybe it's the sum of Gaussians. I'm going to enforce that these 20 variables that I was talking about have a certain distribution. And now I can view this whole thing probabilistically. Right here, we're trying to predict Z from X, and here we're trying to predict X from Z. All right? Now, to make a diffusion network, I just cascade hundreds of these together. And here's maybe what it looks like. Instead of having a latent space that's smaller than the input size, in this version, it's going to be identical. And we're going to have a number of forward steps, what I'm going to call forward steps in this autoencoder, which are the forward steps are adding noise, adding a little bit of noise, adding a little bit of noise. Everyone in this room can take Python or MATLAB and add a little noise to an image. Maybe Gaussian distributed zero mean. And so those are the forward steps. And then I'm going to have reverse steps. And reverse steps are taking away a little noise, taking away a little noise, taking away a little noise. How will I accomplish that? Easy. Maybe just with a simple neural network like UNet, which you could download and run on your PC tonight. So all I have in this diffusion network is adding a little noise, adding a little noise, adding a little noise until I get complete noise, taking away a little noise, taking away a little noise, taking away a little noise until I get something. And the only part where you're going to get freaked out is when you make your own noise. Basically, let's say you train it on 5 million faces, and then you make your own noise and reverse it, and it turns into a face you've never seen before. And that's when, I don't know, that's when you call your sister and exclaim. So that's the diffusion network, and that's really all there is to it. Now, in our problem, we wanted to know, can we do it on ultrasound? Can we do it with multiplicative noise? Which I don't know if you're comfortable with that term. Signal dependent is another way. And what does it look like? Yes, the answer. Thank you, little guy. What does it look like? Instead of adding noise, we're going to add noise that's also multiplied with the signal. And the forward process is going to be easy. We can express it in the logarithmic domain by adding noise to the logarithm of the original image I0. And so that's going to be fine, and we prove that that's Gaussian. The trick is the reverse process, taking away a little noise. And what we found, and this is published on arXiv right now, we're hoping to publish it in IEEE. This is an IEEE talk tonight. What we found is that the reverse process also was Gaussian with known variance but unknown mean. And so here's our idea. We enforce minimizing a distance between the inverse of the forward process and the reverse process. How do we do that? By making their distributions agree. How do we do that? Kullback-Leibler distance by minimizing the KL divergence. And so we make a loss function, and every neural networks paper has a twist on the loss function, and then of course every neural networks paper exceeds the previous one by like 0.5% or something. That's sarcasm. So we make a loss function that depends on this KL divergence, and basically what we found is that with this new model, we can rapidly denoise the image, whereas it took hundreds of steps. You remember this adding noise, subtracting noise. In the traditional additive model, So it was really successful computationally. And let me show you some qualitative results on ultrasound images. Here are some ultrasound images, vessels, cysts, and essentially these are diffusion results, which it seems like the sonographers and the doctors are appreciating. Numerically, basically, we're comparing to traditional methods. This is a partial differential equation method. These are traditional denoising methods. These are more recent neural network methods, AI type methods, machine learning methods. Here's our method here. As we get worse in noise, the alpha makes the noise more drastic, if you will. We're outperforming the other models in both signal to noise ratio and dB. You all are familiar with that. And then also we're maximizing what's called the structural similarity measure, which was developed by my advisor. And once he passes away, he'll probably still get 100,000 more citations on that paper. Just because it's just always cited. So we're doing well in structural similarity, and we're doing well in image quality. And if you're a reviewer, give us some grace. Let our paper go through. No, I'm just teasing. I want to tell you about one other thing we're doing. This is a work in progress. The ultrasound transducer, yes, it is sensitive to speckle. And speckle comes from sub-resolution scatterers that are natural. But it also is basically deforming the image, if you will, distorting the image, if you will, based on the point spread function. And so we'd also like to include in a diffusion network, and we have some successful initial results. We haven't published these yet or submitted them yet, where we're actually learning the point spread function of the transducer along with the multiplicative noise and enhancing images and sharpening them at the same time. And this is also work done by the same PhD student, Shomi. And Shomi, when I met her, she worked for Budweiser, and she's been an awesome PhD student. So here's where we're saying we want to estimate this point spread function as well as denoise the speckled image, which ultrasound people would be very offended if I said that because they don't consider speckled to be noise, which is fair to those people. So that's one thing we're working on right now. Now I'm going into my sort of general part. There's going to be plenty of time for questions. I want to tell you we're living in a really strange era right now. Well, for many reasons. But when I was in elementary school, they told me, Nature, Science, New England Journal of Medicine, those were the big journals. Those were the important journals. Fast forward to just 10 years ago, they're still the big journals. Same ones as, in the 1970s when I was in elementary school. So things haven't changed. But look at last Christmas. Google Scholar metrics for the top journals. Some weird things are happening. IEEE CVPR slipping into the 2 spot. What? That's a conference done by the IEEE. NeurIPS at 7. These are basically, in terms of the most cited journals or conferences in the world. ICLR, and there's more lurking beneath that. PAMI, which is an IEEE journal. And I'm just telling you, for the first time in modern history, computer science, electrical, and computer engineering journals and conferences are popping into this elite crowd, for better or for worse. So it's a strange time. What can we do? What can we tell our students to work on? What can we tell our groups to work on? I want to explain 3 things real quickly, and then end with my last word on AI. And I'm positive and negative on AI. It just depends when you catch me. All right. So think about the first AI successes. You watch Good Will Hunting on Netflix, and then you watch, I don't know, some other movie about Boston, and then we suggest another movie about Boston as the third movie to watch, right? Those data are not noisy. Those data are perfect. We knew exactly the movies you watched when you watched them. Medical data, we're here at one of the world's greatest medical research facilities. Medical data they aren't like that. There's very few of them in comparison to the number of Netflix subscribers and Netflix records. They're noisy, they're not perfect, and they're not all labeled. So we need people to be working on simulation data. And I really think, and we're doing this for our bacteria studies, which I didn't talk about tonight, but essentially people that can make good simulators, and that's based on physics and electrical engineering, in my opinion. People can do good simulators, can really advance the field. And that is particularly true of medicine, which is very important in Rochester. Another one that's important in Rochester is causality. We didn't talk about the transformer tonight, but the transformer basically is the T in ChatGPT. A transformer basically forms massive correlation matrices. It tells you that when a word is used here and then another word is used here, it usually means that the word has this particular context after looking at billions of words. That doesn't tell you anything about causality. So causality, think about you discover a phenotype, something physical that has manifest. Is that the risk factor or is that the symptom of something else? Current AI doesn't know. And so people can work on causality, causal neural networks, and make a big impact. I realize that people are doing that, but it's probably 1% or 1/10 of 1% of the work that's going on right now. So the ability to not only correlate things, but to say this caused that is something that AI in general lacks, and you could work on, maybe tonight. A third one is obvious, especially in Rochester, Minnesota. We will never trust AI. We will never adopt it for medicine if we don't understand it. And that goes back to AI is not a black box. So how do we understand it? We make explainable or interpretable AI. And there's really right now no universally accepted measure of such explainability. We're working on a little bit in our FUS, our focused ultrasound work, but essentially this is a key set of problems. And once again, there are people working on this, but we need thousands of people working on this to achieve success in various domains, especially in medicine. And so explainability is something I think is ripe for the taking. So I've already covered that AI isn't a black box. I also want to, inspired by a book that came out of Princeton, I also want you to not accept AI snake oil. Don't buy AI snake oil. By the way, I've been in meetings with business people, and they're very eager to buy snake oil. So what is snake oil? Snake oil is AI that doesn't work. It doesn't deliver. And let me give you some examples, partial and full. Okay, content moderation. We've all probably used Facebook or whatever, one of these social media. It has content moderation. And what the authors of AI snake oil, which I'm going to put up the reference at the end, these authors from Princeton, what they would say is, yeah, content moderation works pretty well if you're in the United States speaking English. But there's 190 countries that use Facebook, and content moderation doesn't work there. It doesn't translate. So it's sort of content moderation is a partial failure. It's partially hyped. Generative AI, these authors would say it works. And I would say, and this but may be changing, but let's not mistake it for human creativity. It's taking midpoints between things that humans have created. Like my wife is an artist, and if you took a midpoint between her drawing of a dog and my drawing of a dog, which would be horrible, yeah, you would get a new piece of art. Does that create anything? I don't know. We could argue about that. Predictive AI, and you're going to have to read the book. They would say it doesn't work. They would say that AI is really good at predicting the past, but is horrible at predicting the future. Why? Because of you all, humans. We're sort of unpredictable. And I'll give you an example on that. If we look at a graph, something like generative AI could be here. It could be autocomplete. When I use my iPhone, it tries to predict the next word that I'm going to say. And guess what? It does pretty good. And guess what? When it makes a mistake, I'm not going to cry. It's not the end of the world. So this is an example. Well, yeah, it works. GPT sort of works. And when it doesn't, it's benign. I mean, it's not the end of the world. Content moderation, sometimes works, is a little bit overhyped. But if it doesn't work, could it be harmful? Maybe, maybe not. If you're in one of those other 189 countries, you're not going to depend on it. But this is an example which is both snake oil and harmful. Crime prediction. And this is true. Philadelphia, Atlanta, Oakland all bought these crime prediction systems from AI that are AI driven. And guess what? The crime prediction software predicted crimes in the less affluent areas of the city. And then it directed police to be dispatched where they found more crimes. And that got into a negative feedback loop, which is documented in one of the books I'm going to show you on the last page, where more people went to jail, where communities were over-policed and some were under-policed because the software was telling them to go to various geographic places where people weren't as well off. And so this is harmful to those people, and essentially is what the authors would say is snake oil. I'm going to judge my talk success based on whether you're going to read a book or not. And Audible is acceptable. Because I was listening to Audible on the way down on the shuttle. But the 4 books that I would recommend from the last year these are 4 books I read in the last year. This book is awesome. It's not completely negative. It sounds like it's completely negative on AI. It's actually this gentleman's PhD dissertation. Wouldn't it be cool if you wrote a PhD dissertation and it was a New York Times bestseller? This book, it talks about software that was used